Okay, so this is a story of a man and his peas. Gregor Mendel, a Moravian monk. What does that mean? Moravia is not a country that we have anymore today, but the part of the world that was once called Moravia is now called the Czech Republic. So it was at one time Austria. It has been controlled by Germany. It was part of what was created in the country of Yugoslavia that fell apart, and today it is part of what would be the Czech Republic. So that part of land has been fought over a lot, and different nations have ruled it at various times. But at the time that Gregor Mendel was there, it was called Moravia. And he was a monk and a high school science teacher, so I like him. He taught at a monastic school. So much like this school, there were schools that were run by the state, and then there were schools that were run by the church. This was not during the time of the Protestant Reformation had happened. That's not the case. The Protestant Reformation had happened, but he was teaching at a Catholic school at a monastery, and he lived at the monastery. He was a monk. So his two jobs there were to teach science at the school, and then also to be in charge of the garden. So I'm sure he made his students do experiments in the garden, much like you guys are doing. So you can kind of feel the culture of him in his classroom. And he started studying hereditary traits. And his garden grew a lot of peas. I'm sure it grew different things as well, and the monks didn't just eat peas. But when it came to the section of the garden that was pea plants, he used that as an experimental garden to study hereditary traits. And so he very, very carefully bred pea plants with each other and noted the characteristics of the parents and noted the characteristics of the offspring. And by using very careful techniques, he was able to study hereditary traits. And he was the first one to unlock some laws or some principles about how traits are passed from parents to child. And obviously this isn't true only of pea plants. You also have hereditary traits from your parents. You have some things that make you look like mom and some things that make you look like dad. And certain personality traits are also inherited. And so you are a mixture of mom and dad. But how is it that parts of dad and parts of mom get thrown together into the blender and out pop you? How does that work? Gregor Mendel was the one who started understanding the principles that go on behind that. Okay, some text. So he named these plants generations. He would cross two parents, and he would call them the pea generation, for we would say parental in English, but it was patristic or something like that, Latin. So the father's generation or the parents' generation, and then first filial, the first generation of children, would be the generation that was produced by the cross of the first two individuals. And then he would take siblings of that cross, and he would cross them, which is why he was using pea plants, not people, because that's weird. But he would take siblings, and he would make them have babies. And that works in pea plants. And then the grandchildren were the second filial, so F1 and F2. So the P and then the F1 and the F2. And he would follow a set of traits through 3 generations of plants, which means that one experiment would take him 3 years, because he would grow a crop of peas, and then he would carefully store the seeds that were produced and label which ones came from which plants. And then next year he would plant those seeds and see what the children look like and cross those. So this is a very long-term experiment. This isn't like something that he was able to do in one growing season. Crossing a set of traits took 3 years. So very dedicated to his science. He did a lot of traits, and he repeated them obviously over and over and over again. So he discovered lots of principles of genetics, but it was a life work for him to be doing this. He discovered that traits do not blend, but appear in discrete characteristics, and we call those discrete characteristics genes. So, for example, if your earlobe is attached or free, is a gene. So you can feel your earlobe in a so we tell you, does your earlobe dangle, or does it stick to the side of your head? I have a dangly earlobe. There is a trait for. There is a gene for an attached earlobe, and there's a gene for a dangly earlobe. Well, one parent has a dangly earlobe and one parent has an attached earlobe. Your ear will not be like kind of attached. It'll either be attached or it'll dangle, right? That's one example. Being a tongue curler is a genetic trait. It either curls or it doesn't. It doesn't like kind of curl, right? So traits don't blend. Traits are what they are. Now there are some things that we look at and we see, no, there's a blend there, like skin tone. If your one parent is really dark and one parent is really light, it's possible that you're in the middle, right? But what's happening there, it's not that your parents' skin tones have blended, it's that there's more than one gene for skin tone. And so you're expressing some of the dark genes and some of the light genes, and you look in between. But even that, it's not a blending. It's a discrete switches, but it's just how many of those switches have been thrown. So it's always discrete units. He also noticed that some traits mask other traits. He called those traits dominant, others are recessive. And when we start looking at how to do this, you're going to see that he used letters to represent a gene, and if you use a capital letter, then that's a dominant trait. If it's a lowercase letter, that's a recessive trait. So examples, blonde hair is recessive. So it is a trait that can be covered up by dark hair. And so if you have blonde hair, it's because probably both parents, well not probably, it's because both parents gave you that gene to have blonde hair. Now they don't have to express it, they can be carriers of it. But if you have blonde hair, it's because it came from both sides of your family. If either family side didn't give you blonde hair, then you don't have blonde hair, which is why blonde hair is a minority. And as more and more ethnic groups are mixing and more people are multiracial, blonde hair is becoming less and less and less of something that you see because you have to get blonde hair from both sides of your family to have it show up. So it's that's an example of a recessive trait. A dominant trait would be dark hair, right? And so if either one side of your family gives you a trait for dark hair, you have dark hair. So that's just one of many examples. Okay. So Gregor Mendel figured out that you get two copies of, or every individual has two copies of a trait. You have a gene for hair color from your mom and a gene of hair color from your dad. So you have two copies of that gene, but you will only pass one at a time to your offspring. And so your pair of genes segregate, and you give one at a time to your kids, right? And the other parent of your children gives one gene at a time to your kids, which is why your gene will then have, your child will then have two copies again, one it got from you, one it got from your spouse. So he called that the law of segregation. Segregation means to divide something. And so you have a pair of traits, and that pair of traits is divided when you make gametes, when an individual makes gametes, sperm and egg. And so the gamete only gets one copy of each trait. And you saw that in the lab you did with meiosis, where a cell starts with two copies of every gene, and then it copies those copies, so it actually, for a moment, has four. And then it divides, so now each daughter cell has two traits, has two copies of every gene, and then it divides again. So now there are four daughter cells that each have only one copy of the gene, okay? And that's a demonstration of segregation. Hello? Sure. Brianna, phone for you. The other thing that he noticed was that your traits that you have, the genetic information that you have, doesn't always show up. With the idea of dominance, you have some traits that show up always, all the time, that are dominant, and some traits that are recessive that can be hidden. So you could have a dominant trait and a recessive trait in your genes and only show the dominant trait. And nobody knows looking at you that you have the recessive trait. So he developed some terminology to talk about the actual genes that you have in your cells, and he called that your genotype, the type of genes that you have. And then he said what you look like, what is being expressed to the outside world, is your phenotype. And he called it that because it's the type of phenomenon that we see. It's what we interact with. And so phenotype is what is expressed. Genotype is what you have. And so those are some vocabulary terms that he developed. So let's look at some pictures of this. An example would be if you crossed a parent that is homozygous, I don't know that word yet, has 2 copies of the dominant trait for flower color in a pea plant, which is a purple flower. So he used a P for purple, and a capital P means it's got the dominant trait. And this font, it's hard to tell big P's from little p's, but this one has 2 copies of the recessive trait, 2 copies of the gene that can be covered up, and in pea plants, that's a white flower. So if you took a 2 copy of the dominant trait parent and a 2 copy of the recessive trait parent, and you asked them to please have babies, then the first generation, the F1 generation, is all purple. There's all purple flowers. That's because the dominant traits that it got from one parent completely cover up the recessive trait that it got from the other. But then, because these are plants and not people, you ask the brothers and sisters to have babies, and now the white flower shows up again 1 out of 4 times. And he would see this trait all the time, where you have a trait and a trait, you cross them and only one shows up. You cross those and the other one shows up 25% of the time. And so it's an example of one of Gregor Mendel's experiments. And this is something that he saw a whole bunch of times, so he had to kind of describe and explain how does this occur. And so we have more terminology here, and I'll pass out another piece of paper that we're going to use in a moment. So more vocabulary. There's a lot of vocabulary here. There you have 2 copies of a trait. One you got from mom, one you got from dad. Maybe both copies of that trait give you the same information, especially if your family is Asian. Asian people tend to have a very uniform gene pool, where a lot of the genes that you get are the same copies. Mom gave you the same information that dad gave you. Other ethnicities have more diversity in their gene pool. But we call those different forms of a trait an allele. So there are multiple gene types for eye color. There's lots of different eye colors out there. And so there's a gene for a brown eye, and there's a gene for a blue eye, and there's a gene for a green eye. And then on top of that, there are genes for how much of that color is made, how much brown, green, and blue. And each of those are alleles. There's a brown eye allele, a blue eye allele, a green eye allele. Okay? So an allele is the form of a gene. And so we would say that you have different kinds of alleles if you have 2 different pieces of information for a given trait. Okay? And then if both copies of your allele are the same, then we would describe that as homozygous. Homo means the same, and you know that from a lot of examples. Okay? Homo means the same. Zygos refers to the conception cell, the cell where the sperm and the egg came together. That's called a zygote. And they're saying that the zygote had same information. That's what the word means, okay? Homozygous means both parents gave you the same information, okay? Heterozygous means I got one trait, I got one allele from dad and one allele from mom, and they aren't the same, they're different. So that same second part of the word, zygos. First one now, hetero means different. So I got different traits, okay? So in reality, most of us are homozygous for some things and heterozygous for others. Because sometimes mom and dad told you the same thing, sometimes mom and dad told you different things. And nobody is purely homozygous, nobody is purely heterozygous. Depends on how many traits you're looking at. So I'm going to do an example here. There, you have two pieces of paper on your desk. One I gave you first, and the second one I gave you second. Right? That was really hard to figure out. The first one does not have any chemicals in it. It's just a piece of paper. The second one has a chemical that some people can taste. So we're going to do a little demonstration here. We're going to find out who is expressing which allele. So there's the allele for tasting, and there's the allele for not tasting. First, we're going to put the first piece of paper in your mouth, and you're going to like suck on it for a second and just be like, yeah, that's paper. If the second piece of paper tastes like the first piece of paper, then you are not a taster. If the second piece of paper tastes very different, you are a taster. Okay? So just so before we do this, the dominant trait is to taste. So most of you will be able to taste the chemical in the second piece of paper. The recessive trait is to not taste. So it means if you can't taste it, that means neither of your parents will be able, well, they may be carriers, but probably neither of your parents will be able to taste either. So control paper, put the control paper in your mouth first. The first one I gave you, nobody's going to taste anything. Just tastes like paper. Paper. So that's what paper tastes like. Okay.
That's so bitter. I hate the wrong one.
Okay. Okay. Paper. Great. There you go. Now,
I'm kind of scared.
the second one, this will not taste like the other one if you are a taster. Ready, go. So, can anybody not taste it? You don't taste it? You don't taste it?
It doesn't taste bitter.
So the dominant trait is to be able to taste, and most of you express the dominant trait. We have two people who are, quiet please, we have two people who are not tasters, which means those two people are homozygous recessive. They are homozygous recessive because they have two copies of a gene that say you can't taste that. And so they don't have the gene to be able to taste it. The rest of us might be homozygous dominant. We might have two copies of the gene that says, yes, I can taste that. Or you might be heterozygous. You might have one copy of the gene that says you can taste and one copy of the gene that says that you can't. But because tasting is dominant, you can still taste, even though you have a copy of the gene that says, no, you can't. Yes, you can. Okay? So that's just an example of alleles and dominance and recessive.
But how do you not just taste it?
You don't have the chemical, you don't have the receptors on your tongue to be able to pick up that flavor. Yep.
We can't.
So they don't have, so they're like,
Well.
Okay, so let's look back at the same diagram that we had a moment ago. We would then, looking at the same diagram, let's put some correct vocabulary on it. So this parent, I said before, is big P, big P. That's kind of a dumb way of saying it. This parent is homozygous dominant, and this parent is homozygous recessive. This first filial generation is heterozygous. They have one of each trait. And then in the offspring, you have one homozygous dominant offspring. You have one homozygous recessive offspring, which is why it looks like grandma, right? And then you have two heterozygous individuals. And any trait that you look at, as long as it's a simple Mendelian trait, which most of them are, if you were to do this kind of experiment where you have a pure breeding one parent and a pure breeding other parent, in the first generation, they'll all look like only one parent. And in the second generation, 25% of them will look like one of the other parent. Okay? So this is a classic Mendelian example. He did it not just with flowers, but with pea colors and pea shapes and all kinds of things. So Mendel's experiments, he would analyze them by doing something called a Punnett Square. I don't know where the name Punnett came from. Yeah, but he would analyze the progeny that he got from these various experiments by drawing a diagram called a Punnett Square. If you only look at one trait at a time, like flower color or pea color, or is the pea wrinkled, or is the plant tall, or anything like this, then you are doing what's called a monohybrid cross. Mono means one. Hybrid means you're sticking things together. So I've got a parent and another parent, and I'm only looking at one trait. I'm going to cross them, right? Then you call that a monohybrid cross. If you do two traits at a time, it's called a dihybrid cross because you're looking at two traits at a time. So the initial experiments were all monohybrid crosses. He was doing pea color or flower color or something like that. And then after a while, he started doing two traits at a time and analyzing that and doing dihybrid crosses. No matter how many traits you look at, you can predict the ratios of the offspring by using these things. So let me show you some examples here. Here I have a parent that is homozygous dominant for yellow peas. Now when you eat peas, are they yellow or are they green?
Green.
They're green. Green is recessive though. Green is the recessive trait for pea color. And but in America, we decided that peas are green. So most peas in the wild would be yellow. But we have bred out the yellow peas because we don't like yellow peas. They taste exactly the same. And if you go to other parts of the world, their peas are yellow. But in America, we like green peas. So you have a homozygous recessive green pea parent and a homozygous dominant yellow pea parent. And if you cross those two, then look at what happens here. You have big Y, homozygous dominant. You do segregation, first law, and you say, I'm going to give a big Y to these offspring. I'm going to give a big Y to these offspring. So this guy can only give big Ys. All four possible children get big Ys. This parent can only give little Ys. So you take one of their little Ys and you write it here. And you take the other little Y and you write it here. And now you just fill in the middle with what's above and to the left. So this one gets a big and a little, big and little. Heterozygous, they're yellow. This one gets a big and a little. Heterozygous, they're yellow because the big Y covers up the green. Here, same thing. Big, little, heterozygous, they're yellow too. And this one, no surprise, is the same. So 4 out of 4, 100% of the offspring will have yellow peas. So then the next generation, oh, my video's in the way. Let's move my video. Come on, video. There you go. So now I'm going to take two of these offspring, two heterozygotes. And I'm going to take one heterozygous and cross it with another heterozygous. And I have a big Y or a little y coming from this parent. And a big Y or a little y coming from this parent. And so now I have the option for big Y, so now I have a homozygous dominant offspring. 25% of the time it's homozygous dominant. This one and this one get a big Y and a little y. So big Y, little y here, big Y, little y here. These two are heterozygotes. They are still yellow because the dominant covers over the recessive. But this one, 25% of the time, I get a little yellow here and a little yellow here, which means I get a green pea. Now, in American pea farms, we have bred out the yellow gene. So even though it's recessive, all of the peas that are grown in American pea farms are little y, and they will not have a yellow gene. If they were to get a pea plant from some other country, they don't care what their pea color is, then that gene would find its way back into American peas. But we have bred out the yellow gene from American pea farms. So, yeah, so homozygous recessive. So now 25% homozygous dominant, 50% heterozygous, 25% homozygous recessive. Okay? You will be doing lots and lots of these for the next 2 days. Punnett Squares are going to be your life for the next 2 days. And I want to show you just briefly a dihybrid cross. You will get to do one of these as well. But this is where we take a parent that is heterozygous both for pea color and for wrinkly peas. And we cross it with another parent that is homozygous, that is heterozygous for pea color and wrinkly peas, which means both parents will be smooth yellow peas, but both parents also carry the trait for wrinkly green peas, okay? And now you would look at the offspring, and you'll find some that are homozygous dominant for both traits. You will find some that are homozygous recessive, where are you, for both traits. But you will find that some of the time, you have a dominant trait of one and a recessive trait of the other, smooth green peas. And sometimes you will find the other one, the dominant trait of one and the recessive of the other, and have wrinkly yellow peas. But it's still only 1 out of 16 will you find wrinkly green peas, okay? And so the ratio that you always get for this kind of cross is 1 to 2 to 1, 1 to 2 to 1, all the time. The ratio you always get for this, for the phenotype ratio, is 9 to 3 to 3 to 1, all the time. So as you're doing these tomorrow, if you don't get 1 to 2 to 1 and 9 to 3 to 3 to 1, then you did something wrong. And it's a way to check your work and make sure you're understanding what's happening, okay? So we will play with these a lot for the next 2 days. Any questions at this point before I am done? Any questions, any questions? Let's hear it for Moravian monk science teachers.
Yeah.
Okay, here we go. I'm not a Moravian monk, but I still like it.